Microbial examination of milk and milk products. There are three methods: direct microscopic count, standard plate count, dye reduction test. Two dyes we will use, methylene blue and Resazurin. Firstly, we will see the direct microscopic count, that is also known as DMC method. So this method, enables the rapid enumeration of the bacterial cell. In this method, we can see the cell shape and the arrangement also. We can see the number what is present here in the given milk sample. So by this, we can calculate the how many microorganisms are present in the milk, and on the basis of that, we can grade the quality of the milk. So the formula which we are using for the DMC is the DMC, that is direct microscopic count, is equal to N into MF. MF is the microscopic factor. So that is an area of the field that is observed through the microscope. So this MF can be calculated by this formula. So this technique is very useful for screening of the milk supply. Now for requirements for this DMC technique, firstly test tubes with the milk sample. These samples could be raw milk, pasteurized milk, boiled milk, or any other sample. Then hemocytometer and with the cover slip. Then micropipette, methylene blue, and microscope with the oil immersion. Then we will go to the methodology. Firstly, we will prepare the slide. So with the given milk sample, we will make the 0.01 ml of the sample, and that will be on the area of 1 square centimeter. So all this we will do. We will keep this with the help of micropipette on the hemocytometer, and then we will fix the slide. Hemocytometer we are using because on that we can check the field and we can count the number also. So after the fixing of the slide, after the fixing of this film, we will stain it with the methylene blue, and then we will observe it under the microscope. Once we will observe, we can get the number of the microscopic field, and then we can count the number of the cells. Like in one field, how many organisms we can see. These organisms we can see the shapes also, we can see in the form of clumps also. So that way we can count that how many organisms we can visualize in one field. Okay. So the observation that we can do here, this is the hemocytometer in which this is the grid, and we will see this area only first, and here we can see that these are the cells, these are the bacterial cells which are present. We will count from these cells. Okay, so in one field the number of the cells which we can count, that we can put here in the formula, and on the basis of that we can calculate the number of organisms. So this is the formula, firstly to take out the value of MF, that is the microscopic field. After the MF, we can calculate the number of organism, like whatever is the MF and the number of the organism per field, by multiplying it we can get the number of organism. So 1 ml suspension contain like the number of organism we can see, because we need to calculate the number in 1 ml per ml. So this is the grading of milk based on the BIS standard, like what is the quality, like good, fair, poor, or very poor. That is on the basis of the number of organisms present in 1 ml of the sample. Now there are so many advantages of the direct microscopic count. It is rapid method, morphology can also be observed, and various dense suspension can also be counted if the dilution is done properly. One limitation is there, because with the along with the living cell, along with the viable cell, dead cells are also counted. Okay, so that is one of the limitation. So now we will go to the second method, that is the standard plate count. Standard plate count method, again it is for the estimation of the bacterial population. This is again a quantitative method, because we are going to count the number of the cells. Like here DMC is also quantitative, it is also a quantitative method. And in this test we will employ the serial dilutions, like here you can see. After the serial dilution we will plate it, and on the basis of the number of organisms grown after the incubation, we can calculate the number of organisms present in the given milk sample, and on the basis of again then we can grade the milk. Okay, so requirement for this experiment SPC is the milk sample, test tubes, then pipette, petri dishes, burner, incubator, colony counter, and nutrient agar media. So after that we will go for the methodology for this experiment. Here we will make the dilution, serial dilution, then we will make the Petri plates, nutrient agar media will be there, and after that what we will cool down the media, and then we will pour it the media and the sample by the pour plate method on the Petri dish. Then we will keep all the plates for incubation for 48 to 72 hours at 37 degrees centigrade. After incubation, we will observe the growth on the plates. Okay, so then by using the colony counter, we will get the number of the bacteria present in the given sample. Now after this, we can, on the basis of the one more thing is here, like how to calculate the numbers. So like the number of the bacteria, if in this plate, if it is 32 and the dilution is 1 is to 10 to the power of 4, so by multiplying the dilution factor into number of organism observed, we can get the number of bacteria. Like here in this plate, we observe 32 bacteria, 32 cells. So, and the dilution factor is 10 to the power of 4. So, it will be 32 into 10 to the power of 4 number of bacteria here in the this plate. Okay. So, this way we will count the number of bacteria, or the microbial count we can do. Then again, interpretation of the result will be done by this method. Standard plate count less than 2 lakh, it is very good. Then this is the quality, next is the good, then fair, and then poor. Now coming to the advantages of standard plate count, enumeration of only viable cell. Like earlier it was viable and dead cells were also, non-viables were also counted, but here only viable cell counting will be there. Cultural and morphological differentiation based on the colony characteristics. So after the counting, or if we go for the biochemical analysis, we can also see the morphological characterization, and we can identify the organism also. Then suitable for the determination of the quality of milk, useful for the pasteurized. So all these are the advantages. Coming to the third method, that is the dye reduction test. So dye reduction test, again it is based on it is a qualitative analysis, and we will deal with the methylene blue or Resazurin. So here they will act as an oxidation reduction indicator. So what is happening in these dye reduction tests, bacteria consume the dissolved oxygen during their growth in the milk, and consequently they will reduce the OH to a level at which these dyes are reduced. So what is happening here? Here the oxygen is used by the organism, and these dyes are getting reduced, and this will bring the change in the color. Okay, so by that we can see the reduction. Okay, and on the basis of which the time of reduction is taken as a measure of the number of organism in the test. So more the time, lesser the organism, but lesser the time of reduction or lesser the change in the color is more organism. Okay, that we will see here. Firstly, we will see the principle. So the principle for this test is like the oxidation reduction potential of the substance. We know that it is defined generally as the chemical process in which the substance loses or gains the electron. So here what is happening, milk exists like in the when it is coming from the this cow. So at that stage it is sufficiently rho redox potential is there. Okay, but once the oxygen is there during the processing, the redox potential, oxidation reduction potential of the milk will go to 0.3 volt because of the incorporation of the atmospheric oxygen. So at this particular OR potential, methylene blue is in the oxidized state. Now when the bacteria cell multiply in the milk, they will consume the dissolved oxygen and the more oxygen is used, it will get depleted and dye will start acting as electron acceptor. So here now instead of oxygen, dye will act as the electron acceptor, and then what will happen, the oxidation reduction potential will decrease from this 0.06 say to 0.01 volt. So methylene blue will start getting reduced. Okay, so when the number of greater the number of organism in the milk, the greater the metabolic activity and faster is the reduction. So more activity, more fast reduction will be there. Now this was the principle. Now requirement for this experiment is the milk sample, pipette, burner, test tubes, dye, methylene, and dye will be methylene blue and the Resazurin. So these two reduction dye we are going to check for this activity. Now methodology, again it's very simple test in which we are taking the milk sample, in which we are adding both the dyes. So 10 ml of the milk sample in the test tube, and we will add 1 ml of the dyes, whether it is Resazurin, whether it is methylene blue. Then we will mix it properly and keep it in the water bath for incubation at 37.5 degrees centigrade, and note down the time of incubation. Then we will check. Observation will be done after 1 hour. Okay, and after 1 hour we will start checking the change in the color. So and after each 30 minutes then we will take the readings, and then we will observe what has happened to the tubes. Okay, So after 30 minutes, continue to observe the reduction of dye at the interval of every 1 hour. So firstly we will go for the 30 minutes, then we will start taking the observation at 1 hour. So the milk shall be regardless as decolorized when the entire column of the milk is completely, so milk, when we will consider that the milk is decolorized, when complete it is decolorized, or it is decolorized up to the 5 mm of the surface. Okay. So this way the color will be changed after the reduction of the dyes, after keeping them for the incubation. Okay. So here, like with the methylene blue, blue will be converted into white, and for the Resazurin it is the pink color, it will be also be colorless. Now coming to the result. Again, the quality of the raw milk is adjusted by making the following observation. So as we have seen in the principle of this process, that slower, longer the time means quality will be good, lesser the metabolic activity of the microbes, lesser the number, and lesser the time, that means more metabolic activity, that means more organisms are present. So on the basis of MBR time, that is methylene blue reduction or Resazurin reduction time, the quality of the milk can be observed. Okay, so here you can write the dye reduction time. Dye reduction time. Okay, so this dye could be Resazurin or methylene blue. If it is 5 hours and above, the quality of the milk is very good. That means lesser the organisms are there and the metabolic activity are less. And if the time is very less, like less than half an hour if it is coming, the color is going to fade it, or the color is going to disappear. That means the quality is very poor, means more organisms are present. So this way we can grade the quality of the milk, and we can do the qualitative analysis of the given milk sample. Thank you.